So we'll go on to the next talk by Micha Friedman from the School of Chemistry that will tell us about small molecule mediated inhibition of microbial cellular processes. Please, Micha.
Thanks very much. I want to open my talk by breaking a myth. There is no such thing as medicinal chemistry. There's chemistry. When you guys approach a chemist with a question that is related to developing molecules that can give you some sort of biological activity, one may define that as medicinal chemistry. And of course, there are other subdisciplines of chemistry which help us form molecules that turn out to be drugs. But when you approach a medicinal chemist, not sure whether you can find someone specific as long as he knows chemistry. So we know how to make bonds and break bonds, and that helps us design molecules, and that could be attributed to just about any chemical need. And so I think that when you talk about medicinal chemistry, if you ask 100 medicinal chemists, what is it, or what's the definition, you'll find 100 different definitions. But at the end of the day, it's the ability to create molecules with biological activities. And I'm going to show you some examples of what we do. What we are interested in is mainly antimicrobial agents. And I'm going to show you the more basic science part of these projects. And I'll start with one family of molecules, which are called aminoglycosides, and these are these creatures that you see over here. The first one was discovered in 1943 and became one of the first drugs, streptomycin, for TB. So that was quite a breakthrough. And what these molecules do is they bind to this pink site. This is where actually you see a tRNA. This is the A site. This is where the decoding of the RNA is happening. And when aminoglycosides bind there, they actually keep the site on at all time. And this leads to erroneous tRNAs coming in, and you have the wrong amino acid in your protein. At the end of the day, you get enough of this to lead to a damaged proteome, and then the bacteria dies. And so the big problem with aminoglycosides, even though they're very effective, is that they're nephrotoxic and ototoxic. And I can say that till today, over 7 decades of use, there are probably thousands of different aminoglycosides that have been synthesized in trying to reduce the phenomena of toxicity or overcome resistance. However, we try to approach it from a very, well, I would say naive point of view. And what we've done is started looking into natural aminoglycosides and see if we can find some trick to overcome specificity issues. Now, why do we have a problem with toxicity? We have a problem because we have 2 more ribosomes involved, and these are the mitochondrial ribosomes of mammalian cells and also the cytosolic ribosomes. Now they too have an A site. The A site is a little different. And we thought we could use these differences in order to improve selectivity towards the bacterial ribosome. And the idea was, let's add fruitful interactions to the binding site at the A site of the bacterial ribosome, and by doing so, statistically reduce the chances of binding or reduce the affinity to the other A sites which we're not interested in, and by doing so, maybe we will be able to reduce toxicity and improve specificity. And so, luckily for us, in the last couple of decades, there are a lot of crystal structures published. What you see over here are the structures of the aminoglycosides isolated from the whole structure with nucleotides of the ribosomal RNA at the A site. And what we noticed is that one of these families always has a specific hydrogen bond between a ring, which is called the ribose ring, and an amine, which is always positioned on the adjacent sugar. So we noticed that it was there. It was there all the time. It was there for 7 decades, but we happened to notice the phenomena when it comes to one subfamily of aminoglycosides. And that, to us, was a very important phenomena, because looking at aminoglycosides, you can divide them into 2 major families. And so one of the rings over here, I'll just tell you, I'm a carbohydrate chemist, so this is not a carbohydrate. This looks like a sugar, but it's not a sugar. All the rest of them are. And one of the subfamilies is the 4,5 disubstituted 2-dioxystreptamine, which is this moiety. And the other family is the 4,6 disubstituted. And again, what we noticed is that we always have a ribofuranose at position 5 of these aminoglycosides, and we always have a 2-equatorial amine, whereas if we go to the other subfamily, we may have an alcohol over here, and of course we don't have a ribofuranose. Now, looking into these crystal structures, we realize that the ribofuranose contributes a lot to the binding of these molecules, and so what we've done is we thought we could actually merge them and add binding affinity to the A site of the bacteria, and by doing so, again, lose affinity to the other A sites and improve activity. And so we've done that through synthesis. The synthesis is pretty quick on that board, but it took quite a while to be done because we need to do a lot of stereo and chemo-specific chemistries over here to reach a specific position. Anyhow, we ended up with these molecules. On purpose, we've made a few analogs which have a hydroxyl at this position, which obviously should not give us what we want. And we, of course, also generated analogs that have the whole complex as we wanted it. And to cut a long story short, I'm just showing you a couple of examples over here. These are minimal inhibitory concentration experiments that are done on, in this case, on bacteria which are known to cause lung infection in CF patients or are prevalent in lung infections. And what you can see over here is that you start with an aminoglycoside with a certain concentration that either kills or doesn't even kill the bacteria, and you end up with the ribosylated forms where you have a significant improvement in antimicrobial activity. But this is not the reason why we've made these molecules. The reason is specificity, and this slide pretty much summarizes the phenomena. What you want to see is a high red and a low blue. This is what you want to see. Because when you do in vitro translation on eukaryotic ribosomes, which are actually E. coli ribosomes, and you calculate the IC50 value and divide it by the, these are from rabbit reticulocytes, and these are from E. coli ribosomes, and when you divide the values, you want a very high value. So the higher the value, the better your specificity, and as you can see over here, in these compounds which I've shown you that had an improvement in antimicrobial activity, we also had a very nice improvement in selectivity. So that was the proof of concept in this project, but we're now working on the next generations of aminoglycosides, which are probably going to have, or hopefully going to have, clinical value as well. And this brings me to another use that we found for aminoglycosides. Now, what bothers people who design antibiotics is bacteria that do not react to antibiotics. And some of them don't react to antibiotics because of the fact that they're dormant. And if they're not dividing, they don't react to the action of most antibiotics. However, if you rupture the cells of the bacteria, they have to react to it, or they have to die whether they're dormant or not. And this can be achieved by making membrane-disrupting antibiotics. And we thought we could utilize aminoglycosides to do that. And so we start with an aminoglycoside that inhibits a ribosome. We end up with an aminoglycoside that ruptures the membrane. And I'm going to try and persuade you with that in a few slides. And so what we've done is we took an aminoglycoside, and as for why we picked it, it's just because it's a small molecule that has many amines on it. Now, amines are important because they're ammoniums under physiological conditions. Essentially, you can view this part of the molecule as a positively charged ball, and it's a highly positively charged ball, and it's attached to a lipid that would help anchor it to the membrane. Now, since the membrane is made out of many lipids that are held together by bivalent cations, such as calcium or magnesium, when we're going to stick something which is highly positively charged into the membrane, it's going to replace these cations, it's going to pull the lipids towards it and create a hole around it. And so that was the naive idea. And so we've made these molecules, and what we found out is that tails molecules with a tail that has either C12, C14, or C16 chain has the best activity. So these are the lowest MICs that we've got, and the activity was really nice, and we could overcome quite a few bacteria which are resistant to aminoglycoside. At that point, we had no clue that we are succeeding in targeting the membrane, but for those of you who are microbiologists, all of them were gram-positive bacteria, and for a long time, we couldn't target the really tough bacteria, which are gram-negative bacteria, until Raphael, the student who worked on this project, found out that by increasing the hydrophobicity and basicity of the positively charged groups in these molecules, one can actually achieve that. So you start with a molecule that can destroy the membrane of a gram-positive bacteria, but then if you de-N-methylate this molecule, you end up with a molecule that can destroy both the gram-positive and the gram-negative membranes. And if you're asking why is that so, the reason is very naive and simple. You lose specificity by increasing the hydrophobicity, and losing specificity is something that I can tell you is sometimes very useful. So we were very happy to lose specificity in this case, and what was beautiful about it is it was general, is the fact that it was general. No matter which cationic amphiphil we chose, you start with a cationic amphiphil that is good against gram. Can I move to the laser? You start with a cationic amphiphil which is good against gram-positive and not against gram-negative. You end up with one that is good against gram-positive and again gram-negative. And it happens whether it's a pseudo-tetrasaccharide, trisaccharide, disaccharide, etc., etc. So this was a general effect, and it seems to be very useful at destroying both gram-positive and gram-negative bacteria. Didn't tell you anything about the toxicity of these compounds. Someone who dealt with the toxicity is Kfir in my group, who realized that there is a significance to the shape of the lipids in the membrane. Our membranes are more tolerant of unsaturated lipids, cis-unsaturated lipids, whereas those of bacteria and fungi are less. And so he's made this series of compounds, and when testing these compounds, we found out that the more cis unsaturation we enter into the lipid part of the molecule, the less hemolytic activity we get. And hemolytic activity served as a model to see whether we target the membranes of mammalian cells or not. However, a lot of people who work with cationic amphiphils focus on that, on red blood cell hemolysis as the only reason for toxicity, but of course we have many other reasons for toxicity, so we end up checking other cytotoxicity effects, and indeed the same trend is reserved when we work with mammalian cells. So here you see an example of a co-culture of C. albicans, Candida albicans, so these compounds are antifungals, with mammalian cells. And you can see in the control, the Candida cells essentially perturbed all the mammalian cells. You see almost no live mammalian cells in these pictures. When you end up with one of these compounds with one of the cationic amphiphils that is not unsaturated, it is fully saturated, indeed you see way less Candida cells and you see more mammalian cells, but the problem is that these mammalian cells are dead. So these compounds are not selective, but when you increase the degree of unsaturation, you end up with way less fungal cells and live and happy cells. So in terms of dealing with the toxicity of these compounds, increasing the degree of cis unsaturation and the lipids that compose these compounds can lead you to a much better or much more selective activity. We retain activity against fungal cells, but we lose the toxicity towards mammalian cells when we increase the degree of cis unsaturation. Another nice thing that we could do with these molecules is make molecules that you can turn on and off by turning on and off light. And so you can make an antimicrobial agent and in the middle of the lipid install an azobenzene unit, which then allows you to move between cis and trans. And this allows you to turn on and off the activity. And so here you can see this cationic amphiphil with E. coli cells that look, this is the dead live assay, so it's they're lively and they're happy. when the molecule is in this shape, but then when you turn on UV, not with the bacteria in there, because they're going to die from UV, and you add it to the bacteria, then the compound changes its conformation, and then the compound becomes active, so you could activate it with light and then kill the bacteria. And this is an indication of not only killing the bacteria, but also of making holes in the cells of the bacteria, and what you see here is only cell debris. I told you about membrane and the fact that we rupture membranes, but can I show it? There are a lot of assays that we've done to prove that we are targeting the membrane, and one of these you can see over here. Here are Candida cells, a yeast form that express ENO1 that can be found both in the nucleus and in the cytosol, so we use this as a marker. You can see here only the GFP ENO1 picture, and you can see here another cationic amphifield that was developed in our lab. It took a while to find one that can also kill the fungi, but is also inherently fluorescent. So what you see over here is only that compound. And here you see again the ENO1 proteins, and when you overlay, you see that whenever you have a hole in the membrane, you have a high concentration of the compound as well. And what we think that is happening is that regions which are highly negatively charged draw much of the cationic amphifield. We get an emulsion in certain regions of the membrane, and this of course creates a hole, and the whole cell contents is going out of these holes, and this is how it works. I'm going to shift gear. Well, I usually talk in carbohydrate symposium, so I can't talk about it, so I'm using you guys to tell you something about something which is not a carbohydrate. And I'm going to talk about the azole antifungal drugs, and what we've done through a collaboration with the group of Judith Behrman is study where do these drugs go, and I'll tell you in a second why is that important. And so here is some of the things that happen when you have Candida infections. They're amongst the leading or the most common pathogens that cause fungal infections, and what azole drugs do is they inhibit a protein which is a demethylase involved in the biosynthesis of ergosterol, which is essentially the sterol of fungal cells, and has pretty much the same role of cholesterol in our cells. And so this is their target. And when Judith asked me if we could make molecules that would help us see how they enter cells and how they exit cells, I looked into the structures of these molecules, and what we found out is that all of them have this imidazole or triazole ring. All of them have these dihalogenated phenyl rings, but then the third part of these molecules is totally different depending on which molecule you use, and for us it was heaven, because this means that maybe instead of these parts we could attach something which is fluorescent and then find out things about these molecules. And so we've generated two molecules. The true order was generation of these molecules and then docking experiments that were done by Tali from the Blavatnik Center that helped us understand whether we need to invest money and time in making chiral molecules or not. It ended up being that both of these molecules, both the R and the S of these molecules were about the same binding affinities, and so we didn't bother, and we just made the racemic mixtures of these molecules. And here you see the drug fluconazole, and that's the analog that looks like fluconazole. And then other types of azoles like itraconazole and ketoconazole. So we've made the Cy5 analog of these molecules, and you can see that they pretty much overlay, and therefore they both fit. So this analog fits where the itraconazole and ketoconazole fit in the crystal structures of these molecules with the target, and fluconazole and the Denzyl analog fit pretty much the same. And so with that in hand, we just tested to see whether they have the same activity, and indeed we found out that one has the same activity, the Denzyl one has the same activity as fluconazole, and the Cy5 has the same activity as ketoconazole as expected. And when we use a knockout, what we found out is that indeed both compounds lose activity when we have a homozygous knockout of the target, whereas in the other two, we get some residual activity. So that was just something fun to find out, that some of the azole drugs don't really do only one target, they target more than one target. But this is what we waited to do. We waited to see where they go in cells, and what you can see very clearly is that it doesn't really depend on the actual shape, but just having this conserved motif can lead these molecules to the mitochondria. Now that was a big thing to us. I'm going to finish right now. That was a big thing to us because now we realize that azole drugs do not go where they need to go in cells, and that's the ER where their target is actually. And so with that information, and that I'm not going to show now, we are now designing azole drugs which are going better to the ER. One would expect that to lead to a much better local concentration where the target is and better activity. This is what we're doing. We have one analog which is pretty fabulous, I have to say, and of course should or is designed to go to the ER and not to the mitochondria. So maybe the improvement of local concentration at the right position in the cell leads to the better activity of these compounds. So this is the group who did the job, and this is Rafael who did the antifungal activity, and Sivan and Ido who are working on the aminoglycosides, and Kfir who is working on reducing toxicity of cationic amphiphils, and Joanna, my postdoc, who worked on the photoisomerizable cationic amphiphils, and of course for the funding, my collaborators, dear collaborators, and to you guys for your attention. Thank you very much. Well, depending on which type you try. I mean, if there, we were able to make aminoglycosides that are effective against fungi, and we were able to make ones that are effective against bacteria. Again, unfortunately not, or maybe fortunately enough. Yeah.